Surely, at this point, you've seen Imperial Yeast's amazing selection of high-quality brewing yeasts over at imperialyeast.com, but you might not be as familiar with Imperial Yeast's special order yeast bank. In addition to their tried-and-true core strain selection, Imperial Yeast has a number of ale or lager strains, Brettanomyces, Kveik, and other yeasts available for special order. If you're looking to match a yeast strain currently in production, or just looking for something new to play with in the brew house, reach out to Imperial Yeast customer service to see what they may have in store. Special order strains are subject to a 10-liter order minimum and require a 2 to 3 week propagation time, but they might just be what you're looking for your next brew. You can get more information about Imperial Yeast's special order strains by calling 503-477-5826 or emailing customerservice@imperialyeast.com. Welcome to the Brew Lab. One of my favorite things about brewing science is that it's ever evolving. Our understanding of why beers taste the way they do and the biochemistry of what causes those flavors and aromas is constantly changing. Recently, two concepts have had brewers' attention when trying to understand hop aroma and flavor, those being biotransformation and thiols. I'm your host, Cade Job, and today in the lab we're going to talk about both biotransformation and thiols with my former colleague, Ricky Molitor. Ricky is currently a sensory technologist for Boston Beer Company, but he was recently a master's student under Dr. Tom Shellhammer, where he focused his thesis studies on how yeast impacts thiol release in beer through a process called biotransformation. So we'll start the show by talking generally about biotransformation and thiols, and especially thiols-free and bound precursors, but Ricky's research is unique and fun in two ways. First, he partnered with Berkeley Yeast, who created several genetically engineered yeast strains to increase thiol release with only minimal hopping. That means with hopping only early in the kettle. And secondly, Ricky performed detailed sensory analysis to better understand thiol flavor in beer and describe the sensory frequency and intensity of thiol release by these genetically engineered strains. So the research question goes like this. 1, can we genetically engineer yeast to increase biotransformation and release of thiols without negatively impacting other fermentation characteristics? And then, what does the beer taste like? It's going to be a fun episode, and we'll find out the answers to those questions in just a bit. But first, if you still haven't considered becoming a patron of Brewlosophy, what are you waiting for? By becoming a patron, you get unique rewards and you get to select the level of your monthly contribution. The discounts to YakimaValleyHops.com, access to monthly recipes for contributors, and an invite to a monthly live Q&A session. If you're not a part of those sessions, you really are missing out, and it's only $3 a month. These are real all-stars. They're well-known homebrewers, authors, brewmasters, BJCP judges, scientists, podcast hosts, microbiologists, yeast geneticists, beer sensory technologists. It's an incredible wealth of knowledge, which in itself is worth the money, worth the monthly pledge. So this month are the voices behind Basic Brewing Radio. If you're a home brewer and haven't listened to Basic Brewing Radio, well, I mean, you probably have, so I don't know why I'm doing an introduction for them. You know James Spencer and Steve Wilkes, and you've probably listened to that show just as much as I have. The podcast is great. The video series is great. And like I said, these guys from Basic Brewing Radio, James Spencer and Steve Wilkes, will be available to answer all of your questions in this month's Q&A session. And remember, all previous Q&A sessions are available on the private Patreon Facebook page. All you have to do is sign up over at patreon.com/Brewlosophy, and you can become a patron for yourself. Thank you to everyone that's left a rating or review of the show. Please keep it up. I really do read every comment that I can find, and leaving a rating increases our visibility and helps people like you find the show. So if you haven't left a rating or review yet, I'd love to hear from you. Feedback is brought to you by the team at Haas, who are constantly innovating to provide solutions to your brewing problems. We've all had hazy IPAs, and since you're listening to this show, you know that getting a stable haze can be a challenge. Imagine you've brewed this awesome hazy IPA, you added your Whirlpool and dry hops, it's looking great while fermenting, and then drops crystal clear after you crash it. Well, the folks at Haas have you covered. Introducing Hop Haze, their newest brewing product. Hop Haze can be used in post-fermentation to add a stable hazy complexion to beer and other beverages. It's 100% hop-derived, flavorless in beer, and requires no additional mixing before use, and the haze stability is the same as the shelf life of your beer. So if you want to achieve that juicy look in your next hazy IPA, be sure to try Hop Haze, and also check out all of Haas's other innovative products at johnihaas.com. That's John, the letter I, Haas.com. All right, listener Simon wrote in from New Zealand, and he says he has a show idea. He says, "Hey, Cade. Hi, Cade. Enjoying the show as always. I was listening to the Brewlosophy podcast episode that they just did about cold versus hot steeped coffee additions, and I happen to have just finished a keg of black IPA with cold steeped dark grains inspired by your episode on black IPA a while back. I know you preferred the hot mashed dark grains, but man, this cold steeped one was amazing. I wondered about the science of hot versus cold steeping and why the bitterness and astringency seems to not come out in a cold steep coffee or dark grains. There seems to be some other stuff that does come out in the cold steeps, like the tobacco flavor you mentioned, which I totally get from the cold steep wort, but didn't get in the final beer. Is there any science behind this? I'm interested to hear your thoughts." So Simon, yeah, thanks for the awesome show idea. That's a really interesting one. Yeah, my experiment was looking at in a black IPA, what happens if you put your dark grains, if you steep them in cold water the night before versus if you put them directly in the mash. Does that change the actual flavor? And for me, it did, for sure. I definitely noticed a difference, like you mentioned, between the warm steeped and the cold steeped grains, with the cold steeped grain having this weird tobacco-y flavor that just wasn't my preference. I mean, they were both good beers, but it just wasn't my preference. I much preferred the hot steep version. But it's interesting. You seem to like the cold steep for yourself, Simon, and that's awesome. That's why we do this type of experimentation. But it would be really cool to understand the science behind this. So it's a fun show idea. I hope there's some research out there looking into it, and if so, I'll see if I can get them on the show. I think it'd be a lot of fun to look at and figure out exactly what's getting extracted and why these flavor characteristics might be the way that they are. So thanks for the show idea, and I'll see what I can come up with. All right, I'm going to take a short break, and then I'll be back talking with Ricky Molitor about how genetically engineered yeast might improve biotransformation and thiol release. One of the biggest improvements to my brewing practices was the upgrade to stainless steel, and Delta Brewing Systems offers some of the lowest prices on high-quality stainless gear, like the Firm Tank, which holds 8 gallons or 30 liters of wort, comes with a domed lid to reduce the chances of a messy blow-off, and it can hold up to 4 psi of pressure. Delta Brewing Systems also has their own line of brew kettles, as well as one of the lowest-priced all-in-one electric brew systems out there, and their prices are remarkably affordable. If you're in the market for legit stainless gear that won't break the bank, you've got to check out deltabrewingsystems.com. There are hundreds of compounds that contribute to hop flavor and aroma. Some of them exist in high concentrations like myrcene, linalool, geraniol, but there's a class of compounds that typically have low concentrations in beer but are super potent. Those are called thiols. And with me in the lab to talk about biotransformation and release of thiols using genetically engineered yeast is Ricky Molitor, formerly a student of Dr. Tom Shellhammer and now sensory technologist at the Boston Beer Company. So Ricky, welcome to the Brew Lab.
Thanks, Cade. I'm really excited to be here. I will preface, this is my first podcast that I have done. It's evident by how I asked my fiance how I should dress up for this podcast, and then she said, "You're on a podcast. People are just listening to you, so you don't need to worry about what you dress up."
I love that story. That's awesome. Yeah. And you're right. People are just listening. But you'll be able to see a picture of Ricky in the social media. But yeah, it's cool to have you here. And Ricky and I were recently colleagues. So Ricky was a master's student in Dr. Tom Shellhammer's lab, and as Ricky graduated, that's where I slid in as a master's student. And while I did my post-baccalaureate, Ricky was the boss. Ricky was the boss of the hops lab where I got to do some work there. So Ricky and I have a really good friendly relationship. We're former colleagues. And the cool thing today about this podcast is we get to talk about thiols and biotransformation, which I think might be two of the hottest buzzwords in brewing science right now. And we get to talk about both in this podcast. That's pretty cool, right, Ricky?
Yes, definitely. It was an honor to work on these projects, and the timing just couldn't have been better. Like these thiol biotransformation has just blown up. So it's been it's been awesome.
Yeah, exactly. Well, and so you got your master's in food science from Oregon State University with Dr. Tom Shellhammer doing this research on thiols and biotransformation. So what we're going to talk about is one of the aspects of that, which was done with genetically modified organisms. But also the point of getting the master's in food science was also to get a job, which you've done. And you are now a sensory technologist at the Boston Beer Company. So I'll ask two questions because I know you're in the Beyond Beer area, and Boston Beer has had some recent product developments that are pretty cool in that area. But I always ask this question to the guests, and so I want to start here. It's how did you become interested in brewing science?
Yes, I love that question because it gets me fired up. I think it gets a lot of other people fired up. Mm-hmm. Basically, I grew up in Central Oregon, about an hour north of Bend. And as I was growing up, it was an incredible opportunity just to be involved in the blow up of that craft beer scene.
Yeah.
And not just the beer itself, but like the community and the culture that it brought everybody in together. And seeing everybody come to one place and just like tasting these products that brings a smile to their face. So that was kind of the first part. The next part, I'm a little bit of a science nerd. I think everyone in brewing is a little bit of a science nerd.
Yeah.
But I think we're in one of the only industries out there that involves both science and art. And there's like this balance between the two of them. So it's like creating this product, being innovative, but then it's also this science backbone to it. So I think that's a really beautiful thing. And then that got me hooked, started the fermentation science program at Oregon State. From day one there, I knew I was going to stay in this industry for the rest of my life. So cool stuff. I love it. I'm fired up about it.
Yeah, of course. And you were an undergraduate worker in Tom's lab, and then ultimately got into the master's program and then led the hops lab for a year, which is pretty cool. And like I said, now you're at Boston Beer as a sensory technologist. And I know you work in the Beyond Beer area. So why don't you tell us about a little bit of that? And then, specifically two big news pieces that just came out of Boston Beer. So they've released the Truly vodka hard seltzer line, and also Loma Vista, which is a tequila-based ready-to-drink cocktail. So that's a pretty cool area, right? Working Beyond Beer.
Yeah. So the thing that I love about my job right now is like I get to work in all of these little aspects of the company, and that's one of the pros of working for a company like the Boston Beer Company. That's like really trying to innovate and push our industry in all these different directions. So in terms of working in the Beyond Beer category, I know some people in the brewing industry are like, how could you make seltzers, right? But at the end of the day, kind of like what I said earlier, like we're here to make good products that bring a smile to people's face and that people like to drink. So that's what really inspires me in that space. And then there's a lot of cool opportunity to play with a lot of new innovations within that space as well. And even lean on some of the learnings from brewing, or vice versa, learn from what is going on in this Beyond Beer category and bringing that into the beer space. So a lot of cool stuff. It's been a cool opportunity.
Yeah, exactly. I mean, and you get exposed to sort of cutting edge, right? I mean, what's the, you get to be right there at the table talking about what the next big thing in beer might be, right? And I think that's a really cool place to be. And especially at a company like the Boston Beer Company. And again, speaking of cutting edge, I mean, the point of what we're talking about today is how you can use genetically engineered yeast to potentially increase biotransformation and release thiols during fermentation. And so that was one of the big things that you looked at in this study is determine whether you can actually do that. Can you actually, modify a yeast genome in order to get it to release increased thiols? How does that affect fermentation performance? How does it affect chemistry? And then, of course, my favorite aspect of this is also what's the sensory impact? What do thiols taste like? What does this beer what is it? And I think that was a lot of fun for me, especially because I was on the tasting panel for this. And so actually tasting what these individual thiols might mean was a lot of fun. I'm sure you had fun organizing those panels too.
Oh, yes. There were some interesting beers that came out of this.
Yeah, for sure. And of course, this was in partnership with Berkeley Yeast. I think it was primarily Jeremy Roop and Charles Denby that were working on this. But I've had Nick Harris on the podcast before. We've talked about some of their exploits into genetically engineered yeast. And so I guess, can you give me a big takeaway? Did it work? Can you genetically engineer yeast to increase thiol release?
Yes. Our big takeaway here, you can use genetically modified yeast to make really tropical beer with thiol levels up to 73-fold or 73 times higher than their sensory detectable levels. So
wow.
a really big boost in these the amount of these thiols in these beers without impacting, like equally important, without impacting your fermentation performance or beer chemistry.
Yeah. And I love that, right? I mean, because that is important, right? I mean, people think about, oh yeah, you're releasing thiols, you're getting all this character, but if it messes up other aspects of fermentation, that's a problem, right? If you modify the yeast and it acts in different ways, that's a problem, right? You have to make sure that you still have the fermentation performance that you need. And so that's an aspect of it. And we'll get to those results eventually. But I think first we've got to start with talking about some of the basics of hop aroma. Does that sound like a reasonable place to start?
I think that's a great place to start.
All right, well, let's go there then. So let's talk about hop aroma.
Yeah, so when we think about hop flavor and aroma, the first thing we typically think of is this essential oil fraction within the hop cone itself. And this only accounts for about 0.5 to 3% by weight of the hop cone.
Yeah.
Now, what's unique about this is within that essential oil fraction, some recent research has identified that there's up to 1,000 different compounds that each contribute uniquely to beer's flavor and aroma. And of course, we all know this when we're brewing ourselves. If I add this hop variety, it's going to cause the beer to have this flavor or this aroma. Mm-hmm. But up to 1,000, there's a lot of different compounds within that essential oil fraction. So instead of naming off all 1,000 aromatic compounds,
we'd be here for a while.
We'd be here. This would be a very long podcast. We can break those down into three broad categories. So we have our hydrocarbon compounds, and these account for around 64%. These are our compounds like myrcene, humulene. And then we have our oxygenated compounds, which are about 35% of that essential oil fraction. And this is compounds like linalool, geraniol. And then we have this tiny little sliver within that essential oil fraction that accounts for about 1%. And these are our sulfur-containing compounds. And that'll be kind of the target of what we're talking about today, is this one tiny little bit of this hop essential oil fraction.
That's interesting. So this hop essential oil fraction is when you're talking about the weight of hops, it's 0.5% to 3%. So it's already a small amount of like the weight of the hop, but it's super potent. It's super aromatic, right? And then you can go even a step further and break it down into these three classes, like hydrocarbons and oxygenated compounds and sulfur compounds. But then if I did the math right in my head, that means that like 99% of the hop essential oil fraction is these oxygenated and hydrocarbon compounds like geraniol, linalool, myrcene, humulene. And it's just 1% of already, potentially 0.5% of the hop that are sulfur compounds. I mean, we're talking about like really tiny concentrations, right?
Yes, definitely. And that's where within these sulfur compounds this is where we'll first really talk about like thiols. Like what is a thiol? I remember the first time I heard that word, I went, what are they even talking about?
Right. Yeah, me too.
So in a broad sense, a thiol is a sulfur containing compound. And there is an abundance of different thiols within that 1%. So I don't want us to forget about this when we jump into more specifically looking at we'll talk about 2 thiols in particular, but there are a lot more within that 1%, which is even more mind-blowing when you're thinking about the level of these compounds within beer.
Yeah.
And some of those main thiols, so like the major ones that have been the focus of a lot of research, are 4-MMP, we may have heard, or 4-mercaptophentyl-pentantuone. I won't say these again, just given the full name now.
Yeah.
3-MH, or 3-mercaptohexane-1-all, and then 3-MHA, or 3-mercaptohexanol.
Yeah.
So these are kind of the 3 ones that have been the key focus of a lot of beer research, and that's because they contribute to a lot of these tropical aromas. So we're talking guava, grapefruit, passion fruit, and so on.
Mm-hmm. Yeah, and that's it is interesting too, because I've had I think I had Keith Villa on the podcast at one point talking about another thiol, which is 3MBT, and that's a nasty one, right? The skunky light struck hop character. But these thiols are desirable. 4MMP, 3MH, 3MHA, those are the ones that are responsible for these tropical fruity characters that are so desirable in these, especially like new age IPAs. And I guess we should make one more note before going on. Like Ricky said, we're not going to go through the names every time, because again, this podcast would be 2 or 3 hours long if we called it by its official name every time. But there was also a more confusing name change too that happened. So Mercapto, so we'll call it 3MH and 3MHA because that's what most people know. That M stands for Mercapto, which is now sulfanyl. So you might see in literature or published papers 3SH or 3SHA. Those are the same things. It's just a name change to sort of, I think, more easily and more accurately describe the nature of the molecule. So for Ricky and I, we'll stick with 3MH and 3MHA and 4MMP, but just a note out there for the uber science nerds, we're not intentionally being vague. It's just those are what most people are using, and so we'll use those. But all right, Ricky, so then that's some basics of thiols, but what makes thiols so special? If they're at such low concentrations, how do they have such an outsized role in on flavor and aroma?
Yeah, I love this question, because we took this up to 1,000 different compounds, and we narrowed it down to 1%, and then we narrowed it down to even more. So it's like, how are people even smelling these things, right?
Yeah.
So what makes them unique is that they have extremely low aroma thresholds. So for 3MHA, we're talking 5 nanograms per liter. And for 3MH, we're talking 55 nanograms per liter in beer.
Wow.
So what does that mean?
Yeah. What does that mean? Yeah.
Right. Mm-hmm. Right. So we're not talking parts per billion. We're talking parts per trillion.
Whoa. Okay.
I will not be the first person to use this example. But one way to think about it that I just find mind-blowing is take an Olympic-sized swimming pool. Uh-huh. So over 600,000 gallons of water.
Okay. Yeah.
One drop in that pool would be 1 part per trillion.
Holy moly.
It's hard to even wrap your head around. So when we're talking 5 to 55 nanograms per liter, we're saying 5 to 55 drops of water in an Olympic-sized pool, and we could smell that within beer. If the Olympic-sized pool was beer, of course.
That's amazing. Yeah. Wow. There's an idea, an Olympic-sized swimming pool of beer. Get on that, Boston Beer Company. But no, that's incredible. I mean, that really is mind-blowing to think about, like 5 drops in an Olympic-sized swimming pool, and humans can detect it, right? Humans can smell it. Because again, that's what the aroma threshold is. It's where humans can detect it. That's really, really amazing. So these compounds, even though they're in really low concentrations, they're really, really, really potent. This is also interesting too, because I was talking with Tom Nielsen, for the president of the HRC, about some research that HRC is doing into hop breeding to express thiol character. And it's really interesting to think about. Hops have these compounds in them, but a lot of the time the compounds are bound up, I guess, right? Is that like, talk to us about that a little bit, like free versus bound thiols?
Yeah, so I think in terms of like exploring where thiols come from and like how they make their way in beer, right? And this idea of these free versus these bound thiols. So
yeah,
I think the easiest way to look into it is to look at the wine industry.
Okay.
Because they've been studying thiols for decades. And this is, like you mentioned, a hot topic right now. So this is one of the newer things in brewing that we've been looking at. But again, the wine industry has been studying this for a very long time. Mm-hmm. And how this came about was you can take a wine like Sauvignon Blanc, which has these tropical aromas. And they actually have 3MH, 3MHA, and 4MMP in them. Where the question comes up is grapes don't have these compounds.
Oh.
So where along the way is this wine picking up these compounds? And that's where it's easy to picture this idea of biotransformation. So within the grapes, they don't have these thiols, but they have them in their precursor form.
Oh.
And this is when they're bound to another molecule. And that actually makes them completely odorless.
Oh, interesting. Okay.
And that's where this term bound thiol comes into play. So I will make a note. It's technically not the most correct way to say it. These would be thiol precursors. But for simplicity's sake, we'll call them bound thiols.
Yeah, yeah.
And what the wine industry discovered was, hey, when we ferment this out, we're actually releasing these free thiols or these unbound thiols from their bound form. And that's this idea of biotransformation, right? Using yeast to generate these compounds.
Yeah.
And now when we look at the beer industry and like how do they make their way in beer? So hops actually have these free forms of these thiols. So we think about varieties like Sabro or Mosaic, for example. Like we know when we add them to beer, we're going to get some tropical aroma out of it.
Yeah.
But some recent research has actually found an enormous amount of these precursors that are still left within these hops, which to us represents this untapped resource of this tropical flavor potential that's not fully getting used by just dry hopping or adding hops in the brewing process.
That's exactly what Tom Nielsen said, right? I mean, he was saying, that in hops, a lot of hop varieties have free thiols, right? Have thiols in their aromatic form, and it's kind of like playing with house money because you've got Sabro is a great example that it's going to have free thiols in pretty high concentrations, and so that's one of the reasons why it smells nice and fruity and tropical. But then there, so we kind of have a leg up over the winemakers in that sense, right? And that's kind of what he was saying is it's like, all right, you've got all this free thiols, so you've got a whole bunch of house money that you're playing with. And one of the reasons why HRC and the Hop Research Group are focusing on breeding hops that have thiol character in the free form. But this is where biotransformation comes in. And again, this is the point of what we're talking about today is these precursors, the thiol precursors, the bound thiols. So are there hop varieties that have more of the precursors or, versus free? And does that ratio vary dependent on the variety?
Yeah. So again, some more recent research. A lot of this stuff that we're talking about today is relatively new within the scientific community. Some work done by, I believe, Dagon and Schoenberger looked at different free thiol levels as well as precursor levels across several different hop varieties, across several different times of harvest. And essentially they found this wide range of free thiol levels across several different varieties. So yes, it does depend on the hop variety. And I think a good example, we can look at Cascade, for example, where their free thiol levels are detected below 10 nanograms per gram. So relatively low compared to Sabro, which was detected almost up to 100 nanograms per gram.
Okay. Mm-hmm.
So really low levels in that Cascade, below 10 nanograms per gram. But they also looked at the precursors. Uh-huh. And they found within that Cascade that they had almost up to 25,000 nanograms per gram of these precursors.
Whoa, holy moly, that's a crap load. So does that mean like if you can get access to these precursors that you might turn Cascade, which is normally a grapefruit, bomb, it's very, very prominent in its grapefruit character, you might give it more tropical character if you can access these untapped precursors?
Exactly. And that's where this idea of biotransformation really shines.
Okay, I see. And so how does yeast impact biotransformation? So how does the yeast get in there and release these compounds?
Yeah, so the pathway in which 3-MH finally becomes its last in its form of 3-MH, so like the free form we talked about earlier, there is quite a complex pathway. But right before it becomes 3-MH, it's bound to cysteine. So I mentioned earlier, it's bound to something, which is making it odorless.
Yeah.
So this cysteine-bound 3-MH is released by an enzyme, which is a carbon sulfur lyase, also referred to as beta lyase if you're in some of the literature, which will actually release this 3-MH from its bound and odorless form. And I do want to call out here, 3-MHA, we will be talking about that today too. That's not released from the same enzyme. So it is still part of that 3-MH pathway, but it's derived from 3-MH through alcohol acetyltransferase enzyme. So, so different enzymes at play there. But that beta lyase, that's the key enzyme that's really driving that cleavage of 3-MH from its bound and odorless form.
So that's like to say, so cysteine bound 3-MH, and again, cysteine's an amino acid. So cysteine bound 3-MH it gets acted upon by this enzyme beta lyase to release the 3MH from its bound form, and then 3MH then gets converted through a transferase into 3MHA.
Yep.
Okay,
exactly.
cool. All right. Well, so what determines that? I mean, do all yeast do this? I mean, do all yeast have this pathway that cleaves this cysteine and 3MH bond and releases all this tropical aroma? I'm guessing no, because then we would have a whole bunch of tropical beers, right?
Yeah, that's a good question. I can't give you the exact number of yeast. There was a recent study that looked at it. There's this gene that's been a specific target within a lot of these studies. This is this IRC7 gene.
Yeah, so talk to me about that. Yeah,
this gene encodes for that beta lyase.
Okay.
So if the yeast have that present in its genome, it can create this enzyme. And I want to say it's about, no, I won't give a number because
yeah, fair
I
enough.
don't know exactly. I don't want to confuse anyone. But even yeast, it's a very small proportion of yeast that actually have this enzyme, brewing yeast.
Okay, yeah.
And even those that have it are extremely inefficient at converting that bound thiol into its free form. So,
yeah. So that's kind of like that's a gene expression topic, right? You've got the gene, but even though the gene is present, it's just not being used all that much. So there's not a whole lot of beta lyase activity, even in the yeasts that have that gene.
Exactly.
Cool. And so, okay, I think I'm starting to piece the puzzle together here then. So if you wanted to genetically manipulate yeast to increase thiol expression, you'd want to increase the gene expression of IRC7.
Exactly.
Nice. So is that what you did?
Yeah. So the idea behind it, and this kind of leads into our experiment, is we want to take that gene or a similar gene that encodes for that beta lyase enzyme from another bacteria, for example, and then we can essentially integrate that into the yeast strain itself, which would allow it to encode for this enzyme and essentially release more of these thiols.
I see. So that makes a lot of sense. And I don't want to get too far into the weeds on like the exact specifics of, genetic engineering, because I did talk about that with Nick Harris, and again, that wasn't really your focus of the yeast. That was more Jeremy and Charles, the ones who are actually manipulating the yeast. But what's important for us as brewers here is then we've manipulated the yeast. You've added additional, let's call it, additional opportunities for yeast to use the IRC7 gene, right? So there's going to be a lot more expression of beta lyase, a lot more beta lyase activity, and potentially then, or at least the theory behind the experiment is that means that we're going to have a lot more released thiol character.
Exactly.
Yeah. So why don't you sort of briefly walk us through the experimental design in terms of like what beer you brewed and what you did in order to get thiol character out of it, and then we'll take a quick break and then hop into the results.
Yeah, so we took a parent strain. So we wanted to have a control, so something that wasn't genetically modified and wasn't known to encode for this enzyme. So we used a London ale strain called BYL, mm-hmm. and then we integrated these carbon sulfur lyases into the genome of 4 of these strains. So we have 4 treatments here. And then we also additionally encoded them with alcohol acetyl transferases derived from E. coli and Malus pumila. And this was to further drive that 3MH to 3MHA that I was talking about earlier.
I see. Yeah.
So in total, we have 5 strains. And then what we did was we brewed a very low hop beer with those 5 strains. So this is our light hopping treatment. This was simply a kettle addition at 1.5 grams per liter.
Okay.
And then we wanted to see what would happen if we continued to hop. How would these beers smell? What would be the chemistry behind them? So we took one of the strain, with the parent strain and one of the genetically modified strains. We did that light hopping, and then we did a whirlpool addition on top of that at 3 grams per liter.
Okay.
And then our last treatment, we took that parent strain, we did the light hopping addition, the whirlpool addition, and then we did a 10 gram per liter dry hop addition. So we wanted to really push the hops in that parent strain so we could make some comparisons between these genetically modified strains at these really low rates compared to a beer that was brewed with a lot of hops without a genetically modified strain.
Okay, so if I understand this correctly, then you've got like your control essentially is just like, you know, a light hopped, you know, parent strain. But essentially what you did was you brewed a beer with only a bittering addition, right? Like no aroma or mid boil or late hop addition or anything like that. No whirlpool and no dry hop addition. No other kettle hops. And you expected to get hop flavor out of that beer.
Exactly, from these genetically modified strains.
All right, cool. And then of course, you know, if you do a whirlpool addition and then you add dry hops to a normal strain of yeast, you'd expect to get a whole lot of hop character. And so did you compare those beers, like the dry hopped and whirlpool addition, to the beers that didn't even have you know, whirlpool or aroma hopping additions?
Exactly. And we did that with sensory analysis, which I know we're going to get into, and I'm really excited about that. We also looked at the chemistry of those different treatments as well, with two objectives in mind, right? We're trying to increase 3MH and 3MHA without altering the fermentation performance and chemistry. And then we want to see how these beers actually smell. So that's where the sensory piece comes into play.
All right. Well, I'm excited to see how a beer that only has a bittering addition tastes or smells anything like a hoppy beer. And of course, I already know the answers because I was part of the panel. But we're going to take a quick break and then we'll be back and share the results with everyone else. We all know that designing recipes is really fun, and doing it well is so much easier with good software. We at Brewlosophy recently made the switch to Brewfather, and honestly, y'all, we could not be happier. Brewfather utilizes the latest technology to bring you the most robust cloud-based recipe design software that can be accessed anywhere, on your phone, tablet, desktop, and even offline. It also works seamlessly with numerous third-party devices to make it easier to monitor every step of your brew. I know change can be difficult, but trust me when I say you need to go to brewfather.app today to try it out for yourself. That's brewfather.app.
Established in 1995, More Beer has been consistently serving the greater brewing community since the times IPA was expected to be bitter and clear, and there are reasons they've stuck around so long. In addition to their massive product selection and excellent customer service, More Beer has locations on both the West and the East Coast of the United States, which translates into fast shipping times regardless of where you live. And when you spend more than $59, shipping is free. When you're in need of brewing ingredients and gear, there's no better option than morebeer.com, one of the most trusted shops on the planet.
Moving to the Pacific Northwest has really increased my familiarity with IPAs. In Texas, there's usually 4 to 5 pale lagers on tap, and with one IPA and maybe one, and I'll put it in air quotes, dark beer. But here in the Pacific Northwest, it's usually 4 or 5 IPAs and a pale ale and a dark beer. But Ricky, I wanted to ask you this question, especially since you grew up in Madras, which is where Mecca Grade Malt is at, and so close to Bend in the Pacific Northwest. Do you miss Pacific Northwest IPAs?
I have to be very careful with my answer here.
Yeah.
But yes, I definitely miss some good West Coast style IPAs, Pacific Northwest IPAs. On the other side, there are some great beers out here. So it's been fun to really play around, like tasting different interpretations of the IPA itself. I think it's funny because I had a friend, a colleague at work, fly back to the West Coast for something and went out and tasted. They went to some of my favorite breweries. And they come back, and I went, "Oh, how was it?" And they're like, "Oh, it was okay." And I'm like, fair enough, because that's what I said to all of their recommendations. So,
right. Yeah. It's so funny how regional taste sort of really influences things, especially here in the United States. And I'm sure it's like that as well in other countries. But yeah, I certainly have a higher appreciation for Pacific Northwest, but man, I'm still a fizzy lager kind of guy. But anyway, so again, IPAs, and especially new age IPAs, and, East Coast IPAs particularly, with these high hopping rates, high whirlpool additions, and high dry hop additions are known for their tropical and fruity flavors. And one aspect of that, as we've learned, is their thiol concentration. And again, so you're using genetically modified yeast to upregulate this IRC7 gene to release all of this bound thiol character from potentially, from hops with even just a light kettle hopping addition, and then also checking it out in the whirlpool and in dry hop and comparing those against a control. But the first question that you wanted to answer is, does this change to the yeast DNA affect fermentation and chemistry? So let's start maybe there. Were there any impacts to fermentation performance?
And the quick and easy answer to that is no. So all of the yeast performed exactly like the control through all aspects. So lag phase, time to attenuation, and total fermentation time. So,
and it is super easy, right, to say that no. And I think that's a really cool aspect of this. It bears repeating, right? Again, I think a lot of the criticisms of GMO and genetically engineered yeast are, well, you're modifying this organism. You have no idea what impact that's going to have. And when I talked to Nick Harris about it at Berkeley Yeast, he said, no, I know exactly what impact it's going to have because I'm picking a specific gene. I'm not like changing the yeast metabolism of the yeast, or I'm not changing any of these other pathways. I'm just increasing the amount of production of this one yeast. And so it is really cool to see in practice, making a modification didn't impact any of these other fermentation kinetics. Now, of course, there could always be other ways that it's changed, but that's why we look at some of these other chemistry analyses to check, the quality of the yeast and how it performed in terms of fermentation with some more objective results. So fermentation kinetics, all the same. What about chemistry?
Same answer, not no, but very similar right. Results in terms of the chemistry. So we looked at, of course, final gravity, ABV, real extract, apparent and real degree of fermentation. We looked into BUs, which of course varied as we varied our hopping rates, pH, as well as the color, and all of those were falling within that normal acceptable range for the beer. So.
Yeah. And I mean, as an example out there, so like the control strain, the parent strain was 4.9 ABV, and all of the samples were within 4.7 to 5.1, so plus or minus 0.2 ABV. So I mean that's tight, and again probably not a statistical difference. And then even pH didn't change, color was very similar. Fermentation, real degree of fermentation, apparent degree of fermentation, final gravity, all this stuff was tight. So again, no impact it seems on fermentation and chemistry, right?
Exactly, which is what again, like you said, we initially hypothesized, but it's still one of those things when you're doing any kind of genetic engineering work to go back and check, like, are we still making beer? And the answer here is yes, we are still making beer.
Yeah. And of course, the ultimate question is, are we making thiol beer? Are we making beer that's fruity and tropical? And so this is where we'll spend the biggest amount of time of the results section, because again, I mean, this is part of the biggest, report or the biggest results of the project is, can you genetically modify yeast to increase thiol release, and how does that look against its parent strain? So let's so how about we start there? How did the yeast perform in terms of thiol release?
Yeah, so compared to the parent strains or control, right? All of those thiol levels fell below what we could quantify. So we weren't able to actually detect how many thiols were in there because they were so low.
In the parent strain. So the parent strain beer, there were no, and again, just the one that's lightly hopped, you weren't even able to detect thiol levels.
Nope.
Wow. That's amazing.
And the Whirlpool edition, same thing. So even as we increased our hopping rate within that, we're still falling below the detectable level. The only point where we actually start to see some thiols was within that parent strain that was dry hopped at 10 grams per liter. So again, a pretty hefty dry hop with these hops. And that came just above our detection threshold at around 100 nanograms per liter. So
wow,
really low levels within these parent strains.
Yeah, that's pretty amazing that well, and I mean, but you would think too, with Whirlpool edition or with the dry hop edition, you would have increased some of the thiol character, but only with the Whirlpool strain and the dry hop did you actually get with the dry hop, did you actually get quantifiable thiol character with respect to those thiols that you were looking at? That's pretty impressive. And then so for the genetically modified strains, let's talk first about the ones that were just lightly hopped. Thiols in those?
Yes. So the really exciting part. So again, all of those parent strains fell below that detection threshold, which was 50 nanograms per liter.
Wow.
And again, that kind of highlights too, not to get on a tangent, just how difficult it is to analyze these compounds. Picture, again, trying to measure, like how would you measure one drop in an Olympic-sized pool? I love that analogy.
Yeah, right.
Like, it's just extremely difficult to do. That my one call out there. Let's look at 989. This is the one that really stood out. So we had free thiol levels all the way up to almost 4,500 nanograms per liter. So keeping in mind that the sensory detectable levels are from 5 to 55, and we hit almost 4,500 nanograms per liter within this strain.
Jeez, that's crazy. That's crazy. That's like that's so much. That's so much more. I think you said it was somewhere around like 73-fold change, right?
Exactly. Yeah. So just dramatically increase the thiols, and we really did within all of these strains. So we look across the board, we see values of around 3,000 nanograms per liter for some of the strains in terms of that 3MH, all the way up to, again, around 4,500 nanograms per liter. And then looking at the 3MHA, which definitely had much lower levels, but again, it's part of that pathway. And that's another call out of how difficult this research is. If 3MH leads to 3MHA, how do you really quantify how much 3MH you're generating with these strains when some of it could be getting converted to 3MHA? So again, just like really, really complex stuff in terms of studying.
Yeah, that makes a lot of sense, right? Because if you're just quantifying 3MH, then you're missing 3MHA because it may be getting turned into, like I said, 3MHA. 3MH might be getting turned into 3MHA. So you got to kind of look at both of those. And I mean, they do 3MH and 3MHA have different sensory profiles?
Yeah, each contribute uniquely to, again, that tropical aroma. I believe 3MHA kind of can tend to lead more towards this grapefruit.
Okay.
And then 3MH more towards this passion fruit guava note to it. Actually,
okay.
I might have those backwards, Cade.
Well, that's okay. We'll get there in a second. But I just wanted to ask that question. So it is. I was going to ask it in the terms of like, is it stacking aroma? Like if you've got 3MH and 3MHA, does that mean that tropical aroma is going to increase in intensity? Or perhaps I shouldn't ask that question because we have sensory results where you actually asked panelists very similar questions. And again, I was part of the panel on this, so I'll share a little bit of my experiences here when I can. But let's start then talking about the sensory aspects of this. Or wait, before we do that, let's wrap up. So the fermentation performance and beer chemistry unaffected, right? Like no difference or very little differences, probably not even statistically significant differences from the modified strains and the parent strain, but huge difference in the thiol concentration between the parent strain and the genetically modified strain, like 73 times as much thiol production in some of the modified strains than the parent strain, which is just incredible. So we've seen that the yeast is not affecting fermentation. It is releasing thiols. And like you said, the second part of the panel of the research is, okay, what does this mean? What does it taste like? What do these thiols taste like? So walk us through a little bit of the methodology and then we'll get into the results.
Yeah, so we used two different sensory analysis approaches. We used what's called a check all that apply, often called CADA, and then we used a line scaling exercise. We had 18 trained assessors, and we trained them on all of the attributes that we used within CADA, as well as with line scaling. So I can get into CADA. You want to start with CADA?
Yeah, let's start with CADA.
Okay, great. So how CADA or check all that apply works, it works exactly how it sounds. So our assessors, again, they were trained, are asked to smell each of these samples and then check from a list of attributes. So we had tropical, guava, passion fruit, but we also had other attributes such as herbal, citrus, sweaty. So they'll taste these beers, and if they're picking up that compound, they'll select, yes, I'm detecting this compound. And what we can ultimately do is look at the frequency at which each of these beers is getting called out for each of these attributes. And when we look at which of these attributes came out as the most intense or being called out the most frequently, sorry, we can't use the word intense here, but we will get into that with the line scaling.
Yeah.
We see that tropical is our most used attribute, which isn't surprising. We really push those thiol levels. And we see some significant increases in the frequency that all of these genetically modified strains, again, at this really low hopping rate, are being called out as tropical, guava, passion fruit. And then we look at some of these other attributes that are being used to describe the parent strains at these different hopping rates. And we're seeing these more sweaty, fruity, resinous, herbal qualities that are pulling those apart. So
I see.
Really, really interesting stuff there. So again, getting maybe more of that general hop characteristic with those non-genetically modified, but definitely seeing an increase in this tropical passion fruit qualities within those genetically modified strains.
Yeah, and that's really interesting, right? From a sensory perspective, that's what you would hope to see when you're using thiols, right? I mean, you'd hope to see some tropical passion fruit, guava flavors, and that's actually what had highest frequency. I mean, I think tropical and passion fruit were two identifiers that panelists used to specifically identify the genetically modified strains, right? I mean, they were significant to strains that had that IRC7 gene upregulated, which is really cool to see. And again, it just sort of lends more credence to the fact that, yeah, this genetically modified strain can be used to increase thiol character, this tropical and passion fruit character. And I'll say, as I was one of the panelists for this, there are a lot of choices, right? I mean, I don't remember exactly how many attributes that you picked from or that we had on the list, Ricky, but we're talking about all kinds of different things like herbal and citrus and mango and fruity and sweaty and resinous and onion garlic and, all these other flavors that typically define hop aroma. And passion fruit and tropical came out as really, really high, as high frequencies for these genetically modified yeast. And I know for me personally, that's what I got a lot of. I got a lot of tropical. I got a lot of passion fruit. I got a lot of pog juice, I think, which is what we used as a sensory standard, right? For those of us that know passion, orange, and guava juice, that's what we used as a sensory standard. And it really, really popped. And again, remember, the only hops that are added on these beers was a 1.5 gram per liter addition. It's basically just a boil kettle addition, just for some bitterness. I think that's incredible. I think that's a really cool takeaway from this research.
Yeah, it's really cool. And that kind of leads into the line scaling here too. It's like, no duh, these beers smell tropical and they smell like passion fruit. When we brought the thiol level up to, like we said, 73-fold, it's sensory detectable levels. But what we wanted to look at next was, all right, what Cade tells us is how these beers are being described, the frequency at which they're being described. So we can say that they're statistically more tropical and more passion fruit. Mm-hmm. But what that doesn't allow us to do is quantify by how much, how intense is that tropical aroma? Like, they're being described as tropical, but how intense? And that kind of leads into what line scaling is.
Yeah, let's just talk about line scaling then.
Yeah, so essentially, we evaluated all of these beers based on just two attributes. So this was tropical aroma intensity, and then the next one was overall hop aroma intensity. And I do want to be clear, like, it is understandable. Tropical aroma is technically a hop aroma itself. Mm-hmm. One could classify it that way. But for this study, we broke those apart specifically. So for the hop aroma intensity standard, we used essentially just hop oil.
Yeah. Mm-hmm.
And then you probably remember, Cade.
Yeah.
And then for the tropical aroma intensity, we actually used pog juice. So pineapple, orange, guava, for those that aren't familiar, because we really wanted to get an idea of the differences between those two. So yeah, and how this works is you'll taste your beer sample. There will be a line scale from no tropical aroma or no hop aroma. And then on the other side of the scale, it's extreme tropical aroma or extreme hop aroma. Mm-hmm. And again, these panelists were trained on to where to put samples within this line, not these specific samples, but with training standards. Mm-hmm. And then you scale based on the intensity of it. So again, getting into that intensity piece instead of just describing them as passion fruit or tropical.
Yeah, and I can say as a taster in this panel too, right? I mean, he's right. Ricky's right that tropical aroma certainly overlaps with hop aroma, right? I mean, a tropical is a characteristic of hop. I mean, we've talked with Jeff Daly about, hop sensory standards, and tropical is definitely one of the key flavors from hops. But this is the tropical juice aroma, like that passion fruit or that guava, that orange juice that's like very distinctly juicy, is something to me that came through a lot, especially in the genetically modified yeast. Whereas like hop aroma, like Ricky said, is a hop oil, and so there's this resinous aspect to it. There's like I'm not going to say leafy because that's not necessarily the characteristics that I'm thinking of, but it's really that resinous. It's like the smell of hops. You got a bag of hops, you open it up and you smell it and you go, oh, that's hops. It's that characteristic, right? That's hop aroma. And interestingly, I don't want to win your results, but I think that plays a pretty key role for me in describing the difference between the genetically modified beers and the non-genetically modified beer, is this difference between tropical aroma intensity and hop aroma intensity. So Ricky, why don't you walk us through what how the panelists felt about those two different intensity metrics?
Yeah, so we'll start with tropical aroma intensity. No surprise, similar to what we saw with the Cade, we do see some significant increases in tropical aroma intensity for our genetically modified strains, especially compared to all compared to these parent strains. But what's interesting here is we did see some pretty large differences in the amount of thiols that were generated by each of these GM strains. So as I mentioned, some were around 3,000 nanograms per liter, some all the way up to almost 4,500 nanograms per liter. So big differences there, but we weren't seeing significant differences across any of the genetically modified strains.
Huh, interesting. So it's like you got so much hop, you got so much thiols that it was just like, it's overwhelming. It was just like, so they're tropical thiol bombs, right?
Exactly, which indicates there's potentially this level that you hit where you're no longer increasing the thiol aroma by adding more of it. It's not perceivable to the drinker. So that was one of the really interesting findings out of this.
Yeah. And then what about when you compare it to the parent strain?
The tropical aroma intensity or the hop aroma intensity?
No, tropical aroma intensity. When you compare it to the parent strain, did the parent strain show similar tropical aroma intensity?
Oh, no. Significantly lower tropical aroma intensity. So
right.
There was one interesting call out with the one of the parent strains that was a Whirlpool edition. And then one of the genetically modified strains, we weren't seeing a significant difference there. But in terms of the values themselves, you can clearly see the tropical aroma intensity for these parent strains fall well below what they are in these genetically modified strains.
So there is some impact from Whirlpool using just normal yeast and normal whirlpooling practices that increases tropical aroma intensity. But if I remember correctly, you also used one of the genetically modified yeasts and Whirlpool added a Whirlpool addition to that too. Did that have increased tropical aroma character, or like stacking the yeast and the Whirlpool, or was that one just already so high that it didn't make much of a aroma intensity difference?
Yeah, so in terms of like the thiol levels between that Whirlpool genetically modified strain and the non-Whirlpool genetically modified strain, we weren't really seeing big differences in the amount of thiols that it was generating. But that Whirlpool genetically modified strain was significantly higher than that parent strain with the Whirlpool addition.
So like as between genetically modified yeast without Whirlpool and parent strain with Whirlpool, those weren't statistically different, so they were similar. But if you Whirlpooled the genetically modified strain, that was different than the parent strain with the Whirlpool.
Yep.
Yeah. Nice.
Really hard to explain that, but exactly. I think you nailed it there. Like and I think it really highlights too, like the complexity of what we're smelling in these beer products. And there's a lot of other interactions besides thiols that are occurring during fermentation and when we add hops, that could be perceived as drinkers as something that might be tropical. So a lot of factors, which again make this a really difficult field to study.
Well, and exactly. And another one of those compounding factors is like you talked about, right? Hop aroma and how tropical flavor and aroma, how tropical aroma is subsumed or a part of hop aroma. So I loved this, that you also did hop aroma intensity, and let's talk through those results.
Yeah. So interestingly, across all the genetically modified strains, as well as all the parent strains, except the dry hopped ones, those were all significantly lower in hop aroma intensity than the dry hopped parent strain. And that one was significantly higher than all of the other strains with all of the other hopping additions, either genetically modified or not. So it took dry hopping at 10 grams per liter to really boost that hop aroma intensity character.
That's amazing. And if I remember too from your Cade data, you said that like the resinous attribute was highest for the parent strain with dry hopping. So that hoppy resinous character, again, that for me was the big differentiator between the Thiol, the genetically modified strains, and the one that was dry hopped, was this hoppy character, right? That resinous aspect.
Yeah, and I think that brings us to another good point, which is, yeah, we boosted the tropical flavor in these beers, but it still may be lacking some of that resinous quality that you might get from adding more hops somewhere along the process. So there's this middle ground, but overall, I think it's this new innovative tool that brewers can use to create these new flavors within beer products.
Yeah. And I mean, I think that was some of my experience as well, too. You and I had discussed it a little bit, that our favorite beer out of the panel was that genetically modified strain that also got the Whirlpool edition. Like, that one was really nice. And even though it didn't have as much hop aroma intensity as the parent strain with the dry hop, to me, that was the gestalt, right? That was the one that I was like, oh man, this is really good with the Whirlpool and the genetically modified strain, because you got all that tropical character, but there was enough of the supporting hop components. And I don't know what supporting components those were, but that was my favorite beer out of the panel.
I would agree. I love that beer.
Yeah, that beer was good. It is so rare that we make an experimental beer that's actually like, I want to drink that, you know?
Exactly.
But yeah, back to the point though, too, I would definitely agree that some of those genetically modified strains that just had the kettle hopping addition were really one-dimensional, right? I mean, it was really like, and I would describe it as tropical fruit juice. It was like a tropical fruit juice beer, not a tropical hop beer, but a tropical fruit juice beer. Almost like you just poured some juice into some beer, which is great, and I liked it. But I do appreciate that complexity. So the cool part about this research is you've shown, hey, yeah, we can use genetically modified or genetically engineered yeast to increase thiol character, but then determining what makes the best beer, there's still a lot of room for brewers to play around here.
Yeah. Again, just another item in the toolbox to just create these new flavors and continue to play and find that balance that just works for your brewery. So it's a cool future. I'm excited to see where it ends up going.
Well, and that's a fun aspect for brewers too, because like you said at the top of the show, right? Beer is equal parts like science and art. And so the science says, hey, yeah, these genetically modified strains will increase tropical character. And the art is in what combination, right? In what combination with what hops and what specific yeast strain are you going to make the best beer possible? And that's what I love. It gives the brewer something to think about and a real tool that they can use, like you said, to play around in their own breweries. I just think that's a lot of fun.
It's so cool. We're living in the future of brewing right now, and it's
I know we really
exciting.
are. We really are, and it's fun too. There's an upcoming episode with Matt Winans where we're going to talk about a hybrid yeast strain, Imperial's Capri, which is another alternative for increasing different aroma and flavor characteristics in beer. But we've got these really cool pathways, right? You got yeast hybrids, you've got genetically modified yeast, and ultimately all of these things can be seamlessly integrated into the brewing process. And so again, some big takeaways from your research. The GM strains they had a 73-fold increase of 3-MHA, also an increase, of 3-MH, there we go, and also an increase of 3-MHA without impacting beer chemistry and fermentation performance, which were minimally impacted, if at all. So again, this is kind of a success story for genetically modified beer and tropical flavor. And the really cool part of this is you managed managed to brew a beer with high tropical aroma intensity without using any hops, which I think is just incredible. I mean, you used some hops as a kettle addition, but you didn't add any flavor additions, any whirlpool additions, any knockout, any dry hops, and you made a beer with high tropical aroma. So I've got to ask the question, what's the next step in Thiol biotransformation research?
Where to even start with that question? Because as I mentioned, the wine industry has been studying a lot of this stuff for decades. And it's just such a complex, I know we touched on it throughout the episode, it's just such a complex field. So I think the next step is just continuously learning about these pathways and how they work. And we looked at 3MH and 3MHA. So like trying to figure out ways of studying, like how can we just look at 3MH production? Or how can we just look at 3MHA? Or why stop at these compounds when we have all this future technology that can allow us to play around with new flavors and aromas? And it's an endless field, and I'm excited to see a lot of where the industry's taking this. Like you mentioned, looking at hybrid yeast strains and all this stuff. So big opportunity for research, big opportunity for brewers to keep creating these beers and exploring.
Yeah, I mean, it sounds like thiol and biotransformation are going to continue to be hot topics in the brewing industry. And I always ask this question at the end of the show. So if you want brewers to take away just one thing from today's episode, what would it be?
Yeah, one thing from this episode. I kind of said this earlier. It's kind of a cheesy answer when I say the future is here nice. With brewing. I would hope that brewers would walk away with this going, wow, there's a lot of potential in this field and a lot of opportunity for our brewers to continue to enhance our products, to tying this all the way back to the beginning, put a smile on people's faces when they drink it. So understanding that this technology is really powerful, we should be careful with it, and we should, again, continue to learn more about it. But just be willing to accept this and like go along for the ride and make some fun beer.
Yeah, make some fun beer, make some people happy drinking it. I love that. I love the tie back to the top of the show and why you got interested in doing brewing science. So again, Ricky, thanks for doing this research into thiols and biotransformation. And of course, I mean, thanks to the Berkeley Yeast folks also for their work in actually making the yeast strains. And is there anything else that you wanted to talk about that we didn't get to today?
I think we covered the bulk of it. Again, excited to see where the industry goes. Thanks for having me. It was fun.
Yeah, of course. It was a pleasure to talk to you. I love talking about thiols and biotransformation, and really good luck as you explore other beyond beer beverages at the Boston Beer Company. So Ricky, thanks for joining me in the Brew Lab.
Thank you. Great first podcast.
Yes, you did fantastic. You did great. All right, well, listeners, in the show notes, you'll find a link to Ricky's article titled The Sensorial and Chemical Changes in Beer Brewed with Yeast, Genetically Modified to Release Polyfunctional Thiols from Malt and Hops. This was published in the journal Fermentation in August 2022. If you like what you heard, shoot me an email, Cade@brewlosophy.com, and don't forget to listen to our other show, the Brewlosophy Podcast. The Brew Lab is a production of Brewlosophy, where they who drink beer think beer. Don't forget to visit brewlosophy.com to read about our weekly experiments and other brewing adventures, and listen to us talk about it on our other show, the Brewlosophy Podcast. Thanks to all of our sponsors and patrons that help make this show possible. If you'd like to receive a reward for helping us do what we do, visit patreon.com/brewlosophy to see how you can do just that. Thanks so much for listening. We'll be back in the Brew Lab with another guest next week. Until then, think beer.